So in the last video, we have discussed how the signals are transmitted within the neuron. So we have discussed with the conduction of a nerve impulse within the neuron. So if you remember, we said that if there is a neuron, assume, okay, so one of the neuron, and there is the cyton part, so we have the cyton here. So there is a cyton. So from here at the axon hillock region, we said there will be the setting of the depolarization wave, and the depolarization wave will pass all along this nerve fiber. That's okay, but this depolarization wave will reach the edge of the neuron, which is called as axon terminal. So this is reaching the last part of neuron called as axon terminal. Now the question is how this depolarization wave will pass to the next neuron. For example, we have another neuron waiting here. Assume this is another neuron waiting for the signals, which is in the polarized state, okay, and it is having the dendrites. So you know the dendrites, right? So we have the dendrites here, which can take the signals from other neurons. So how this depolarization wave, which is carried here, which will carry to the axon terminals also, from here how this will go to the next membrane or the next neuronal membrane? That is the question, and that will be answered in this video. That is the physiology of nerve impulse transmission. First of all, we should understand that this junction, it is called as synapse. The junction between two neurons, or the junction where the depolarization wave will pass from one neuron's axon terminal to another neuron dendrite, or to the another muscle, that's called as synapse. So synapse can also be called as neuronal junction. So because the synapse is only between a nerve and a nerve, and this last nerve may lead to muscle, that's not our point. Our point is how these signals will go from one nerve to another nerve, and with the help of a junction between one nerve to another nerve. What is the junction called? The junction is called as synapse, and as it is junctioning the two neurons, it is called as neuronal junction. So how these ions, which has been depolarized, if you remember the last video, if you have looked in the last videos, we have discussed how the sodium ions will be increased inside the axonal plasm or axonal fluid, and they'll reach the terminals. Okay, so now if you zoom this portion out, if you try to just enlarge this view here, if you observe this point, you can see that we are having the membrane. So this is the membrane of the axon terminal, and assume this as the membrane of the dendrite, next neuron. So this is the presynaptic membrane. So what do you call it? It is called as presynaptic membrane, and this can be called as postsynaptic membrane. Why? Because these before the synapse and these after the synapse. So these things have to be understood first of all before discussing how the ions pass out. So the depolarization wave with all the sodium ions will come here. With all the sodium ions, you know, the sodium ions will be more inside when it is depolarized. So this depolarization wave of sodium ions will come here, and this depolarization wave has to pass to the next neuron. So how this happens, that we have to understand. But before understanding, this passing is done in two ways. This depolarization wave is passed to next neuron in two ways, because of the synapse present, because of the type of synapse present. Now what are type of synapses and how this is passed, we will discuss one by one. So first we have to start with the synapse, which is called as electrical synapse. So we have different types of synapses, actually two synapses in our body. The first one is called as electrical synapse. So why it is called electrical synapse? Because here the signals are transmitted electrically in the form of current. in the form of ionic current, if you remember the sodium ions current or a depolarization wave. So there is no requirement of any chemical. They'll directly pass this information or pass the depolarization wave to next neuron. How? So if you observe these electrical synapses, the presynaptic membrane, so this is a presynaptic membrane, will be in very close contact with the postsynaptic membrane. So postsynaptic membrane will be very close to this presynaptic membrane. So these are presynaptic membrane and these are postsynaptic membrane, and they are very close. And if you observe here, these two cells or these two cell membranes, they are connected by cell junctions. If you remember your previous the structure organization of animals lesson, in that you can see that there is some intercellular junctions. So here specifically there is a junction called as gap junctions. So here these two membranes, they are connected by junctions called as, what do you call the junction here? They are called as gap junctions. So gap junctions are a type of junction which will allow free movement of ions. Not only ions, free movement of all the substances, any substance. Why? Because in these gap junctions, you can see here, there is some channel proteins between two membranes. You can see this gap here. So these are called as tunnel proteins. What do you call them? They are called as tunnel proteins, or they are called as channel proteins, or they are called as channel proteins. So these tunnel proteins or the channel proteins, they will be allowing the easy movement of the axoplasm from one neuron to another neuron, or from one cell to another cell. So as there is direct connection of the cytoplasm, you can easily think that these ions which are being present here, the sodium depolarization wave or the sodium ion wave, will easily pass to the next layer or will easily pass to the next neuron, and they will activate the ion channels present on the membranes of this neuron. So they will easily activate these membrane channels present on this neuron, and the depolarization wave sets up in this neuron. So there is not much problem because of the help, thanks to this so-called tunnel proteins or the channel proteins, which are directly sending this depolarization ionic wave or ionic current, we can say, into the next one. Now this type of connections can only be seen in gap junctions. Very important point to remember. The electrical synapse can be seen only in the gap junctions, where there is gap junctions. Without gap junctions, you can't find these electrical signals. It takes help of some other things. Remember, if you remember, we said that these channels are voltage gated channels. So just the ionic current is enough. All the channels on this membrane, which we have discussed previously also, the sodium gated channels or the potassium gated channels, all of them are voltage gated channels. What does that voltage gated channel mean? That means these gates are activated or deactivated with a small fluctuation in the current or in the voltage around them. So it is as simple as that. This transmission is the fastest transmission in our body. So it is the fastest nerve impulse transmission. It is very fast when compared to the next other type of transmission. It is the fastest type of nerve impulse transmission because there is nothing stopping them. So once the depolarization wave sets up, the depolarization will start continuing to the last part where the neural muscle junction will be there. The neuronal muscular junction will be there. We'll discuss that in the coming classes, don't worry. So this is easy. So this is faster. Now you may get a doubt, sir, where is the location of this particular electrical synapse, or in which type of cells or in which type of muscles we can see this electrical synapse. We can find these electrical synapses in areas like the smooth muscles. See, because we require the faster movement of the signals. Where do we require the fast movement of signals? We require them in the involuntary organs, the visceral organs. So we can find these synapses, electrical synapses, generally in the visceral muscles, or we can say smooth muscles, if you remember, smooth muscles. But there is some specification here. These are found only in the single unit visceral muscles. So you could have known this, what is meant by single unit, sir? These muscles, for example, if these is a batch of smooth muscles here, so happy muscles which are waiting for the signals. So these muscles will get activated as one unit. So if this muscle is activated, for example, the neuron gave the signals to this muscle will give signals to all these muscles. So all these muscles will be activated at once as a single unit. Because they are having gap junctions here. You remember the tunnel proteins are the channel proteins. Okay, what do you call the tunnel proteins? They are called as connexons. Those tunnel proteins or channel proteins are called as what? Connexons. So these are connexons here. So these are connexons. So connexons will give the free movement of the ions, and all the muscles are activated as such from this neuron. It should be faster. Now where we can find them? We can find them in the single unit visual muscles. That is, in which parts means in the GI tract, the gastrointestinal tract, okay, and then we can also find them in the uterus. From where I don't know this urethra came from. We have that in the uterus, and we have it also in some other muscles like cardiac muscles. So cardiac muscles also have this gap junctions, or we can say electrical synapse. So the single unit viscera is examples are these two, the gastrointestinal tract and the uterus. Now electrical synapse examples, we can also have the cardiac muscles, which also have this faster transmission of nerve signals because they have to work continuously. So these are some of the examples where we can find electrical synapse, and there is faster movement of the ions, or the easy movement of ions, where they can simply activate without any requirement of chemicals. That is about the electrical synapse. Now coming to the other type of synapse. So generally these very less amounts, because it is only seen in the muscular junctions where we have these smooth muscles, or it is seen in the cardiac muscles, or it is seen in these two batches which we have discussed as the examples of these two. So we can see them only in these regions. We can see electrical synapse only in this region. Now what about the other junctions? What about the other parts of our body? In the other parts of the body, we can see one more type of synapse which is called as chemical synapse. So that is about the electrical synapse. Now we will go through the next synapse called as chemical synapse. So chemical synapse. When you observe this chemical synapse, this chemical, as I said, when you say synapse, we'll have a presynaptic neuron or presynaptic membrane, and we'll have the postsynaptic membrane. So obviously we'll have the presynaptic and the postsynaptic membrane. So this is the presynaptic membrane, and this is the postsynaptic membrane. Now the difference here, then the electrical synapse and this synapse, the difference here is the connection between the two neurons, connection between the two membranes, the presynaptic and the postsynaptic membrane. Here there will be a gap of around 10 to 20 nanometers between two neurons. So there will be a gap of around 10 to 20 nanometers. Now within this gap you can't have the ions to pass out, and there is no gap junctions here. There is no gap junctions so that the ions can easily pass. Now this gap, which is very small, 10 to 20 nanometers, is enough to stop the depolarization wave in this neuron only. Then how the depolarization wave goes to this one? That is the main point. Before discussing how it happens, we should discuss what is present here. What is present in the presynaptic membrane and the postsynaptic membrane. So in the presynaptic membrane of this chemical synapse, we can see a swollen structure. What we can observe is we can observe this swollen structure at the axon terminal. This swollen structure is called as synaptic knob. What do you call it? Synaptic knob. Knob means swollen. So knob means a small, door handle knob, we call it as. So the door handle like that. Okay, so we have synaptic knobs here. And synaptic knobs contain some special packets of chemicals So I'll use some other color so it will be easier for us to remember. So they'll have special packets in its knobs, and these packets are called as synaptic vesicles. So what do you call them? These are called as synaptic vesicles. So they are called as synaptic vesicles. Now what does this synaptic vesicles contain? Synaptic vesicles contains some chemicals. So they are filled up with some chemicals which are called as neurotransmitters. So what we can find here is neurotransmitters which are holded or which are held by this synaptic vesicles. So the neurotransmitters will be ready for their action. Okay, so you can see a lot of them, and each vesicle will measure around 50 nanometers. Diameter will be around 50 nanometers of each vesicle, and this vesicle contain all the neurotransmitters. Now what about the postsynaptic membrane? See, if you remember, in the membranes part we said that we'll have the voltage gated channels. So here we can see the voltage gated channels. So in all these places we can see the voltage gated channels. Remember this point? All these are voltage gated channels. That means these channels are activated when there is change in the voltage or potential difference. But here in this point, in this junction, so in this point, the voltage gated channels won't work because the voltage is not coming out, the potential difference is not coming out, or the ionic current is not passing out. So here there are some special type of channels called as ligand gated channels. These are all sodium channels, remember, because depolarization always depends upon sodium. So here we have special channels called as ligand gated channels only in the postsynaptic membrane. Again, here all we will have the voltage gated channels, but here we have only the ligand gated channels. Specifically, what means sodium? Specifically, sodium. We can see one more type of gated channels which are called as chlorine. So in this region we can see sodium ligand gated channels and we can see chlorine ligand gated channels. Why are they? I will discuss coming video. Okay, so just remember, this is the structure which is present in this chemical synapse, or the presynaptic membrane and the postsynaptic membrane. In the presynaptic membrane we have what? The synaptic knob. Inside the synaptic knob we have the synaptic vesicles, right? And we have the voltage gated channels around here. We have that gated channels here. Here also we have the gated channels, but these gated channels are not normal voltage gated. They are ligand gated channels. What is ligand gated channels? We'll discuss now. So these are about all the structures present here. Now think of this one. What is meant by this ligand gated channel? So what is about this ligand gated channel? Ligand gated channels are the channels which will open up. Ligand gated. Ligand means it requires some support. So ligand gated channel. So to be simple, ligand means activator. So in a simple words we can call it as activator, or we can say helper. So these gated channels which you have observed in the postsynaptic membrane, they require the help of ligands. They require help. So they will be opened up, whether we have discussed the sodium ligand gated channels or chlorine gated channels, they all require the support of some chemical. That is what we have discussed, the neurotransmitters. So neurotransmitters will go that side, they will be released in the gap between the two membranes, and what they will do? They will activate these channels. So we will just put it in a diagram. So think that this is the presynaptic membrane where the depolarization wave came in. So this is the direction of depolarization wave. So here what we can see, here we can see several voltage gated channels. In the presynaptic membrane we can see several voltage gated channels anyway. So we can see here the voltage gated channels. Now remember that here we not only see the sodium voltage gated channels, we don't only see the sodium voltage gated channels, we also see the potassium gated channels here. We can also observe the potassium gated channels. But the main thing here is we can observe the very important point. We can observe the chlorine gated channels, sorry, calcium gated channels, very important point. So here we can see calcium gated channels, Ca2 plus. So we can observe in this area lot of, I should mark this them as calcium gated channels. We can see lot of calcium gated channels. So these are all what? Voltage gated channels. That means they'll be activated when there is voltage difference, right? So now what happens? When the depolarization wave comes here, the calcium gated channels opens up. So generally it is not open, it is closed. They are all closed, like the sodium gated channels. They are all closed. Which one? The calcium things. When this depolarization wave comes in, when this depolarization wave attacks these channels, they will open up and they will have a rapid influx of the calcium ions. They will have very high influx of the calcium ions. Now what will these calcium ions do? So we will have a lot of calcium ions. So a happy boy coming in. So this boy is very happy, calcium anyway. So they came in, and what will they do? What will they do is they will bind with the synaptic vesicles. I am drawing only one vesicle for us to understand. They will bind with the synaptic vesicles, which contain the special chemicals called as neurotransmitters. So these calciums will come and bind with the synaptic vesicle, and what they will do? They will drag this synaptic vesicle to the membrane. So they will make this synaptic vesicle fuse with the membrane, the presynaptic membrane. Then what happens? The chemical, the neurotransmitter is released into this gap. So this neurotransmitter is released into this gap between the presynaptic and postsynaptic membrane. Now we will see. So this is the postsynaptic membrane. Now what do you call this gap here? What do you call the gap between the, what we said, the 10 to 20 nanometers gap? So I said we'll have a gap of between 10 to 20 nanometers. What do you call this gap? This is called as synaptic cleft. What do you call the gap? It is called as synaptic cleft. So the neurotransmitter is released into synaptic cleft. Again, what this neurotransmitter will do? The neurotransmitter will go near the ligand gated channels. So they are closed actually, remember, the ligand gated channels are closed here. So they are closed. Okay, they are closed. Now what will they do? This neurotransmitter will go to this region. They'll attach to these channels. They attach to this ligand gated channels, and they will open up. This will open up. What they'll open up? For the influx of the sodium ions. So these are ligand gated channels, as I have said. So if they go and activate the sodium ligand gated channels, sodium gets in. So we have lots of sodium, which will start the depolarization wave. Is it or not? So now this nerve is also activated. The wave will continue as we have discussed in the previous videos. Now there is some problem here. Why means? Because all the neurotransmitters will not activate the sodium channels. That means all the neurotransmitters are not activating the other neuron. They are not exciting the other neuron. So there is a chance that the other neuron may also be inhibited. So what we have discussed here is the exciting process. So this neuron is exciting the another neuron. The excitation process is shown here. But there is a chance that there are some neurotransmitters which can even suppress the next impulse, which can reduce the next impulse. Okay, that is inhibition process. Now how the inhibition happens, I'll tell you. So assume that this same we have neurotransmitter. I'll give you the examples also afterwards, don't worry. So we have the neurotransmitter here, but these neurotransmitters, what they have done is they have not activated sodium, but what they have activated is they have activated chlorine gated channels. Assume. So what they have done is they have activated the chlorine gated channels. Now assume this neurotransmitter went, So the sodium is still blocked because they are not opening the sodium gated channels. They are still blocked. They are still blocked. So there is no sodium influx. Sodium is staying outside here. But if this one is activating this chlorine gated channels, assume this one. This is a chlorine gated channel. So this ligand gated channel, if it goes and activates this chlorine gated channel, what happens? There will be a rapid influx of what ions? Of the chlorine ions from the ECF here. So it is not the neurotransmitter which goes and activates, it's ions, always ions. So chlorine goes in. Sodium is not activated, chlorine is going in. And what will chlorine do? Chlorine will initiate the hyperpolarization. So they will activate the hyperpolarization part. Do you remember the graph? Remember the graph I will draw here. So this is the minus 70 millivolts where we have the polarization. Then we got the depolarization. Then we have this phase where we got the nerve in resting period. So this phase, what do you call it? This is called as the hyperpolarization. What do you call it? This is called as hyperpolarization, or we can call it as refractory period. So if you remember that word in that video, I said that this is because of the potassium outflux. That means potassium going outside, that's true. But in this case, the chlorine influx can also cause this reduction of the potential difference. So assume we are having potential difference of minus 70 millivolts here. The chlorine came in, negative ions came in. So negative, what it will do, it will convert this minus 70 to minus 90. So this nerve becomes inhibited. That means this action is reduced, not excited. The action is inhibited. So in chemical synapse, we can see both the exciting stimulus and the inhibition depolarization wave also. We have exciting depolarization wave and inhibiting depolarization wave. It's actually not the depolarization wave, it's actually the neurotransmitters which are doing this work. But anyway, it can excite this thing or it can inhibit this thing. Now, how that is happening? Which type of neurotransmitters can activate the sodium gated channels, the exciting part? Which type can activate the chlorine channels, that is the inhibitory part? So we have types in our body. So we have types of neurotransmitters. We have excitatory neurotransmitters and we have inhibitory neurotransmitters. We look at them. So what are the excitatory neurotransmitters? So excitatory neurotransmitters which we can observe, excitatory, are, we have neurotransmitters like acetylcholine, which is an excitatory. We have neurotransmitters called as sympathin, epinephrine, norepinephrine, glutamate. So all these are the neurotransmitters which are exciters. That means what they will do? They will go and activate the sodium ligand gated channels. So they will activate the sodium ligand gated channels, which will carry the depolarization wave to the next neuron. But we have few more things like GABA, gamma aminobutyric acid. If I'm not wrong, I'm just guessing it. Gamma aminobutyric acid, I think so. Maybe it's okay. If it's not right, it's okay. Now we have the GABA there. Then we have some other inhibitory transmitters like serotonin. Then we have the, why I'm forgetting, it's okay. So GABA, serotonin, we have dopamine. I'm sorry. So someone is coming into my mind. So this is coming into my mind. This GABA is very bad. So GABA is actually making me think of other things. So GABA is actually, I'll write the full form here so that my brain becomes normal, because in my brain these are being released. Gamma aminobutyric acid. So this is the actual name of that GABA. So now my brain is happy. My brain is releasing some of the activators like this. Anyway, so GABA, serotonin, dopamine, these are some of the examples of the inhibitory neurotransmitters. These are the inhibitory neurotransmitters which will activate what? This will activate the chlorine channels, which the chlorine will go and produce the hyperpolarization wave, or reduce the potential difference and set to the hyperpolarization, where the nerve will go to the refractory period. So they will stay in the refractory period. Now in this is very important, acetylcholine. Why? Because acetylcholine in the heart, it is on this side, the inhibitory side. So acetylcholine normally in all the other places it is activator or the exciter, but in the heart region this is inhibitor. So that is the neurotransmitters which will either excite the other neuron or inhibit the other neuron, but nonetheless they are transmitting the signals either to excite or the inhibit to the next neuron. So nonetheless they are doing everything. Now my doubt is, if this chemicals stay in that synaptic cleft, think that this is a synaptic cleft. So this is a synaptic cleft here. So if these chemicals stay for longer time, so if these chemicals are staying here for longer time, then how can the next wave come here? How can the next wave come and either excite this or inhibit this? That means here this chemical should not be in for larger time, or we should say longer. I'm somewhat bad in English. So longer time. Okay, so it should not be there here longer time. What happens now, once this chemical is released and once it has activated or inhibited, what happens is there are some chemicals in our body, in the ECF, the extracellular fluid, which will damage these things, which will inactivate them. I'll give one of the example. For this activators, generally for the activators, we have some chemicals called as esterases. What are the chemicals called? Esterases. Okay, acetylcholine, or we can say choline esterases. So choline esterases, what will do? They'll cut this acetylcholine. They will act on this acetylcholine. So I can write somewhere. I'll write here. So I'll rub this up and write here. So what they will do? They will cut this acetylcholine into acetate plus choline. Now this is acting on this. Now this is waste. This cannot activate, or this cannot do the anything to the next ligand gated channels. That's waste of time. So if it's present in this form, that's zero. Like that, esterase can damage all these things. Okay, we have something else like monoamine oxidases. Monoamine oxidase, which will damage the dopamines. Monoamine oxidase. So what will monoamine oxidase do? They will oxidize the amino group in this dopamine, and the dopamine is done. It is wasted. So the cleft is ready for the next signal coming through it, or signal coming to it. So it's somewhat like that. So these things are very, very important. The esterases and the oxidases are very important in reducing the activity of the neurotransmitters. So think that here we are having this, what you can say, cholinesterases. Okay, so now we are having problem. This guy, for example, there is a neuron here. So this neuron is being activated by this cholinesterase. This is very bad now. This is feeling bad. No, I want just to relax. Now what will done? For the rescue comes, no, I'm sorry, this acetylcholine. Anyway, so acetylcholine here. So acetylcholine is continuously poking this fellow. Please become activated. Now he is bad. This guy is bad. Now comes the cholinesterase to the rescue. Now comes the hero, we can say. Cholinesterase comes to rescue, and what will he'll break it. Now stop doing it. He'll break it, and this activation is stopped. Now this guy becomes happy. Obviously he becomes happy. He's ready for the next stimulus. He's ready for the next stimulus to come in. In the same way, if you observe the same point, if there is one more neuron who is also feeling bad, so we are having one more neuron here. This neuron is also feeling bad that he is being inhibited. So this guy, okay, he is feeling bad because he is being inhibited by dopamine, assume. So he is being inhibited, he is being suppressed. No, don't get activated, he is being suppressed and he is not happy. Now who comes there for the rescue? The monoamine oxidases. The monoamine oxidases comes to rescue and they will break this one. Now there is no one acting and this guy becomes happy, ready for the next stimulus. So that's how the stimulus is transmitted. That's how the depolarization wave is transmitted from the one neuron to another neuron. Hope this helps you for the NEET exam in a simple way. Thank you. Mohan Krishna signing off.